My name is Chen. I'll introduce the paper submitted to Multiomics. The title is Multiomics analyses reveal that PRMT5 regulates membrane transport and cholesterol synthesis in white adipocytes. Adipocyte-specific PRMT5 knockout mice showed phenotypes including progressive lipodystrophy, reduced energy expenditure, hyperlipidemia, hepatic steatosis, glucose intolerance, and insulin resistance. To understand how PRMT5 knockout affects transcriptional pathways, we performed RNA sequencing using epididymal adipose tissue via Illumina high-throughput sequencing platform. Functional enrichment analysis of differentially expressed genes showed that metabolic pathways and transport-related pathways were significantly enriched. We next examined whether the observed gene expression changes led to metabolic functional alterations. Using indirect calorimetry, we found that PRMT5 mice exhibited a lower oxygen consumption rate than control mice during both day and night. PRMT5 mice also showed a significantly elevated respiratory exchange ratio compared to control mice, suggesting a metabolic shift from lipid oxidation to carbohydrate utilization. Among the significantly enriched pathways by differentially expressed genes, we found several of them were related to transport functions. Downregulated genes were related with monoatomic ion, iron ion, lipid, and sodium ion transport pathways, and upregulated genes were related with protein, monoatomic ion, peptide, proton, and carbohydrate transport pathways. Loss of PRMT5 leads to a general trend of decrease in lipid transport and increase in cholesterol and glucose transport. We next analyzed how PRMT5 knockout affects glucose transport. Using gene set enrichment assay, we identified glycolysis in the upregulated pathways. We also measured the glycolysis rate and found that extracellular acidification rate associated with glycolysis was significantly higher in knockout mice. Based on functional analysis, fatty acid metabolism pathway was significantly downregulated in PRMT5 knockout mice. In contrast, differentially expressed genes enriched in oxidative phosphorylation pathway showed a trend of being mostly upregulated. Our findings suggest that the loss of PRMT5 inhibits lipid transport and biosynthesis while promoting lipid oxidation. To understand how alterations in lipid metabolic gene expression affect lipid composition, we conducted a mass spectrometry-based lipidomics analysis and found significant increases in the contents of PC and SM. In addition, there was a significant upregulation of genes enriched in cholesterol homeostasis pathways. These results indicate that PRMT5 mice promote cholesterol biosynthesis in WAs and free cholesterol accumulation in the circulation. In summary, PRMT5 knockout downregulates genes related to fatty acid uptake, triacylglycerol biosynthesis, and fatty acid oxidation, while upregulating genes involved in glucose transport and glycolysis. In addition, PRMT5 knockout upregulates genes involved in peroxisomes and cholesterol biogenesis. These changes led to alterations in lipid composition, notably reduced triacylglycerol content and selective increases in glycerophospholipid and sphingolipid species.